Welcome to Akhand Vidyashram, the first institute of impeccable wisdom, embryology, the perfect fetal development. My dharma, impeccable wisdom, my greatest discovery, Divyan, the divine design of nature, with which perfect object of the universe are designed and created. Impeccable wisdom is enlightening, Akhand Vidya wisdom is empowering. Let's understand the science of embryology. Science of embryology is the science of fetal development. Starts with the union of ovum and sperm, starts with the formation of zygote. Zygote takes rest for 1.5 years is an ancient Indian statement which I have repeated. After the formation of zygote, it takes rest for 1.5 days, does not do anything. After that, only a dichotomous division starts. The single unicellular zygote goes through dichotomous division to produce a beautiful multicellular human being. This is a statement which I call the perfect embryology. Now, the different divisions or parts of embryology are the cell division, the cell migration, cell differentiation, self growth and development, the death of defective created cells, the formation of perfect human being. But in this particular thing, we are going to take only mitosis and meiosis only. Then self-development, the human being develops after birth. Many changes occur in many organs of the body. All the tissues, cells, organs, organ systems are essential. All the 10 organ systems should be perfectly normal if you want to lead a perfectly healthy, wealthy, wise, happy, long life. Defects can create many problems and sufferings. Hence, a comprehensive study of science of embryology is mandatory. If we understand what is embryology completely, from living cell to another living cell to the unicellular to multicellular, if A to Z is completely known, we understand biology fully, and then we can be perfectly healthy, wealthy, wise, and happy. And that is where I say embryology is absolutely essential. Now, living cell, I have already told you, there are under compound microscope 3 parts, under electron microscope there are 21 parts, and one part which is centrosome becomes 2 to become 22 part. This is the first stage of creation. Then, of course, the 10 stages of development are there, which consistently continue to be everywhere. But then this important part, all the blueprint for genes for the creation of the every living being, including plants and animals, is embedded in DNA. And B double helix DNA is exactly designed with Deviant ratio, perfect to the core, I have already explained. And there are 22 autosomes of the cells plus one pair of sex chromosomes, X and Y. X is with the female and Y and X are for the male. And you can see Y small. As a result, if somebody understands that if they have impregnation just at the time of ovulation and the person is perfectly healthy, the product, the child is going to be son. If it is before or after ovulation by few hours, then chances of female are much more. And that is also the something which I am giving, how we can decide to have one son, one daughter, so to have a perfectly healthy, balanced life for generations to come. Now let's understand for mitosis, the cell division. Cell division mitosis occurs in normal autosomes only, 22. Each cell divides to form two nuclear cells. The diplomide number of chromosomes, 44 plus XY, or 44 plus XX, are maintained by replication of DNA in mitosis. Each daughter cell has the same number of chromosomes as the mother cell. And this particular process of mitosis takes place in 5 stages. First phase is prophase, and in this the chromosomes of DNA are duplicated. The two identical chromosomes then gradually condense and acquire a rod-like appearance, you can see down, and two centrioles move towards the opposite side to form two poles, and they form microtubules to form the spindle. This is something I have explained earlier also. I am explaining again. You can see the picture down. You have a centrosome which gets into two centrioles. They go to the opposite side, and then the spindle is formed and microtubules to make sure you can see in the second part. Second is the metaphase, mean multiplications. Proposal is the propose, meta is multiple phase. The second division, the nuclear membrane disappears fully. Spindles growing cell is fully formed. Chromosomes move towards the center of the cell, and they are attached to the spindle at the centromere. Please view the growing picture as shown down. You can see very clearly all what I have spoken about it on top. Then anaphase, the third phase. Centromere splits longitudinally so that each chromatid gets separated. They now form an independent chromosome, you can see very clearly. The cell now has a double amount of chromosomes of 46 pairs. 46 plus 46 is 92 chromosomes. You can see in this cell, anaphase stage, they are one together. But now you can see a groove, cleavage forming between the two, which are preparing ground for the next stage to cut it. And that is something which is very, very important. And in telophase being distant, separating from each other, they form two daughter cells with each 46. The nucleus reappears, chromosome gradually become uncoiled. This is followed by division of cytoplasm wherein two daughter cells are formed fully. You can see the picture very clearly what exactly telophase is. They are distancing each other, means becoming far and far away. And cytokinosis is a process when the fifth division, when they divide into two. During this phase, the cell divides into two daughter cells. Intermediate phase called interphase. The daughter cell can go through the phase again. After cytokinase, the cells come to a stage. They go through a phase called interphase. That is consistent between mitosis and meiosis also at the same time. Now, mitosis we have understood very clearly. Now we need to go to stage of understanding meiosis. In meiosis occurs only gametes. That means ovum and sperm cells, the one which produce the human being. Other autosome cells are required to replace the dead cells to prepare the body for maintenance and dissolution. But procreation of a new body between the union of male and female, ovum and sperm, and that process is called meiosis. Now let's understand the definition of meiosis. As I said, occurs only in gametes, ovum and sperms. Two successive division, meiosis 1 and meiosis 2 are two successive divisions. The first meiotic division is a reduction division. Diplomatic number of chromosome is halved to the haploid number during this particular process called meiosis 1. When two gametes with the haploid number of chromosome 23 fuse, the diploid number of chromosome 46 is maintained for good work thereafter. Male and female coming together is what I'm talking about. Now let's understand the interphase. I told you this is the longest phase of the cell life. That is in between two phases. During the phase grows and occupies the DNA. This phase occurs before the prophase state actually starts. This very important phase, you can see how things are taking good shape in this particular picture. Now starting the meiosis 1, which has got the first phase called prophase. And prophase has got further divided into 4 phases. And first is called leptotene, the first division of prophase. In this particular thing, you can see duplication of DNA content. You can see in the picture down. Chromosomes become dense and visible. You can see the visible. Two chromatids are attached to each other as you can see in the picture. However, the chromatids cannot distinguish each other. They look like a pair together. That is leptotene. Second is zygotene, the second division of prophase meiosis 1. Two homologous chromosomes align close to each other and constitute a bivalent status. You can see at the bottom. One out of each homogenic pair is inherited from each parent. Now here you can see one is from father, one is from mother. It is called conjugation synapsis. Each pair comes to lie in the middle of the spindle in zygotome. Now in the third part is pachytene, the third division. Two chromosomes become distinct. They were together, now becoming distinct. Each bivalent node has 4 chromatids, tetrad, 4, 2 central and 2 peripheral. Two central chromosomes become coiled with each other and cross at 38.196 points. The site where chromosomes cross are adherent and chiasmatic. This is again Deviank ratio only. Some people might say between 30 and 40, but I am clear it is 38.1966. If it is a perfect one, it should be designed like that only. You can see in the picture it's like that. Upper portion is 61.8034, lower portion is 38.196. Then fourth division is diplotin in meiosis 1, which is part of propose. Each pair chromosomes move to the opposite side with the spindle contraction. Now each piece is moving to the opposite side. The chromatids involved in crossing over become attached to the opposite chromatid. You can see in the picture very clearly. There is thorough shuffling of genetic material. Each cell produces distinct genetic content. Now, this is what is happening in 4 stages of propose. These are the 4 stages which go hand in hand through the proposal stage to prepare the son for the next generation of meiosis 1's second stage. What is the second stage? It's metaphase. You have seen the moving opposite side become attached to the spindle at the equator. Equator is middle. You can see they're very clearly distinct. But here you can see the changes which are different from what you've seen in propose. Propose both are seeing. Now exchange is taking place. Now you see red part has gone up and blue part has come down. That's a combination which is going to take place in metaphase, multiple divisions. Now next stage is meiosis 1's anaphase. One entire chromosome of each homologous pair separates and moves towards the opposite poles. You can see it moving to the opposite pole. You can see the same combination with a variation. There is no splitting of the centromeres. Chromosomes number become halved or become haploid in the process. That is something to be understood in anaphase, which is part of meiosis 1. Now in telophase, you can see both are separating from each other. Nuclear membrane is formed. This is followed by division of cytoplasm. Two daughter cells with haploid number of chromosomes, 23 are formed and they're separating from each other. They're going from distance from each other. Cytokinesis there is not there. Now comes the second stage of meiosis 2, which is very similar to set mitosis. There is no duplication of DNA. Each chromosome splits at the centrome and chromatids are drawn towards the opposite pole. You can see right on top vision. That is normal meiotic division is the first one. Then further you can see the second meiotic division. You can see the whole thing is divided into 2 each. So 2 have become 4 in the process. But now the genetic material content is not identical in the daughter cells. You can see the division started combinations. And now what you have is 23 single chromosomes, not 46, not 92. That is something which I want everybody to understand is this is how stages are designed and created one by one. Now, let me now take you to a very graphic presentation of the whole meiosis 1 and meiosis 2 in the process so that we are very clear what exactly happens in meiotic division or meiosis division, not mitotic division, meiosis. As I told you very, very clearly, First thing we have to remember is the interface, which is common between two divisions. You can see the cell is duplicating to prepare it for proposed mitotic division or meiosis divisions. But since we are talking about meiosis, let's take interface, the first stage, the cell right on top. You can say the meiosis 1 interface, cell is ready. Now when cell is ready, it goes through the process which you can see is prophase, the proposal phase. In that picture, you can see how they are getting split into two. I'm sure you can. Now let's revise it very clearly. In proposal phase, you find there are one centrosome which has two centrioles which split, and both the centrioles go to the opposite side. They move towards that. When they move to the side, then spindle is formed between these two with the microtubules, and chromatic material in the middle gets up in the equator and the center part. That is the first phase in proposal phase. You can see right on top, you have preparing centrosome to split into two centrioles, go to the opposite side and create a spindle and have the in the middle. That is the first stage of meiosis 1. The second is metaphase. You can see in that chromosomes on the other side, you can see the spindle, you can see the microtubules, but you see the chromatids right in the center, which is there, metaphase. And I'm sure you can understand that this has got 46 chromosomes, and that's it. Then anaphase, you go to the next stage when they start dividing towards one side or the other. Then telophase, they are separating from each other. And when you come to the stage of cytokinesis, the fifth stage of meiosis, which like a mitosis, the two cells are separated from each other, and these two are daughter cells. What was the first stage where all the condensed material started coming to the middle, formed, very ill-defined. Now you got defined, now you have a chromosome in the kind the way you want it, just like any other cell on the whole. That means you have literally come to stage of two daughter cells. Now with the daughter cells, two daughter cells from meiosis 1 go through the further division in meiosis 2, and that also has another 5 stages. That means in meiosis 1 and meiosis 2 there are 10 stages only. Is it a surprise or it's a divine plan? It is a divine plan because everything has to have a consistency so that uniform nature understands this is what it is designed by nature, this is it has to go through. That's what I'm able to see 10 stages everywhere, whether it is structural part in DNA or you have functional part in atoms, elements, molecules, compounds, everywhere, or even functional part in the heart or other part. Everywhere is Divyank, Divyank, Divyank and Divyank. And that is beauty. Now, having understood meiosis 1's interface, which is a common between two phases, then proposed metaphase, anaphase, telophase and cytokinase is one part. Then immediately goes to the meiosis 2 stage. The 2 start becoming 4. I have shown in the last one how 2 become 4. Each one is further divided, so as a result you create different stages. But then you also have same 5 phases. Proper, you can see the similar thing. And metaphase, how they are separating into 2 parts. One has become 2, prepared getting 2. Then in anaphase, you can see this division, cleavage is starting. They're moving to our opposite side. Telophase, they're more further separated from each other. And cytokinase, they become 4. What was started with 1 become 2, 2 become 4. This is how meiosis 1 and meiosis 2 goes in spermatids as well as ovums, and this can be seen in both the things simultaneously. I'm not talking of zygote, I'm talking about sperm and ovum, and these are the things which go very clearly in that. And if this is understood, we understand embryology very well. As a result, what is haploid number of chromosomes, 23 in male, 23 in female, get together to form a zygote with 46 chromosomes, 23 plus 23, and that's where the life starts again for the new child and new creation and beautiful whatsoever together. And now coming to the last part, you can see how we started mitosis in autosomes of 22, which has got 5 stages. Then come to the meiosis 1, which has got 5 stages. Then go to the stage of meiosis 2, another 5 stages. Isn't it amazing that nature has followed the system so well that finally the child which is born out of the union of ovum and sperm is perfectly healthy from every angle, and as the child goes very well, things would be much better. Now that I've explained about menstrual cycle of the female, the perfect one, and also now about embryology, perfect one, and also I've talked about cosmic Purusha as described by Aitri Upanishad, and we also have now modern science of allopathic design. Now next stage I'm going to take to both to show how much more we can learn from the ancient Indian wisdom, or how thousand and thousand years back they were very clear how things are going to be developed, and that has been confirmed today with the modern science. So someone says our people did not have the modern equipments to study. Then how did they give such a beautiful knowledge when it was not there? And that is going to be the next stage. And I'm sure it's going to be very enlightening, empowering, and very useful for the entire universe to create beautiful document on human production. Anyway, thank you very, very